Good morning, biologers. You wanted an introduction, so here it is. It's your boy, Kish. Hiding down in the basement right now since my wife is painting upstairs. The washing machine is running, so hopefully you don't hear too much background noise. All right, so this week we're going to have a couple chances for you to get a couple grades by drawing some phases of mitosis and perhaps even doing a mitotic index. We're going to learn about meiosis. We're going to connect that to Punnett squares and learn how some diseases are passed on through inheritance. And yeah, that'll be it. Let's start off by a little practice right now. I want you to draw me a cell. So we're going to draw a cell in, let's say, prophase of mitosis. And let's make this cell have an N of 4. Ooh, okay. So take a look back at your notes. What happens in prophase? What happens in prophase, and where should cells be? Where should everything be? Okay, so the first thing that you should have is you should have drawn your cell. You should have drawn the nucleus kind of breaking apart, and you should have had your centrioles kind of on opposite sides with some spindle fibers forming. Now the real challenge is in figuring out how many chromosomes there are. So I'm going to pause my video again and draw them while you think about how many there should be. All right, so let's take a look here. So 4, the N of 4 means that you should have 4 unique shapes or sizes. That's what that means. And so in my case, I have little guy, just a little bit bigger, and the biggest one right here. Now, I do need to have 2 colors because remember that they're, I should write that down too. You should have 2 colors, one color representing the maternal set of chromosomes of DNA that was inherited, the other one representing the paternal set. And in prophase, remember that replication has happened. Replication has happened, which means that there should not be 2 of each, meaning 1 black, 1 red of each, but there should be 2 black, 2 red of each. So that amount of DNA should be pretty huge. Okay? Let's practice one more here. Let's take this same set of chromosomes. Can you draw this same cell in anaphase of mitosis? Okay, I'm going to pause my video to write some things down. And you pause this and try to draw this cell here in anaphase. All right, here's my cell in anaphase. Now it looks a little messy, so let me point out a couple of things. First of all, I've got my centrioles on opposite ends of the cells. I've got my spindle fibers connected to each of those chromosomes right at the centromere. And let's see here, let's zoom in a little bit. Let's count up all my chromosomes here. I should have 4 red and 4 black of each shape on each side. And I've got 1, 2, 3, 4 black and 1, 2, 3, 4 red over here. I've got a long one of each. I've got a short one of each. I've got medium ones of each. I had a little trouble connecting the sizes here, but I've got the right number, and that's what's important. And over on the other side, I've got 1, 2, 3, 4 black, 1, 2, 3, 4 red. Okay, and that's the point here. All right, let's shrink back down. Okay, I'm going to erase these things here, and let's get started. on today's notes by doing a little bit of a refresher for when we do mitosis versus when we do meiosis, okay? So let's see here. So let's remember that mitosis creates genetically identical daughter cells. And they are genetically identical to the parent cell. Mitosis is used in repair when you need to replace cells, in development, and this is when the cells will not only divide but also differentiate into a very specialized kind of cell by expressing certain genes on the chromosomes. And when you need to grow and make more cells, that's when you do mitosis. This is when you take a 2N cell and in S phase you double that so it's twice as much. So there's twice as much DNA, I should specifically say. That process is called replication. Remember that replication happens semi-conservatively. And then finally, the cell divides in cytokinesis, and you end up with 2 identical daughter cells to the parent cell that have diploid nuclei. Diploid meaning 2 of each chromosome. Meiosis only happens in one place in your bodies. Meiosis is used in the production of gametes. I believe we've gone over this word, but let me just say it here. Gametes are sex cells. Gametes are sex cells. And in humans, the male gamete is the sperm, and the female gamete are the eggs. And in this one here, let's see, and in meiosis here, what's going to happen is that we are going to start with a 2N cell. After S phase, that is going to be 2N cell with double the amount of DNA. And that is going to divide once into 2 cells, and then each of those is going to divide again. And so we're going to end up with 4 total cells. Now we're going to draw a very generic meiosis. This particular process isn't exactly how eggs are formed. So we are going to just draw a very, very, very generic version of meiosis. But the point in meiosis is that each of these cells that we make only has 1 of each set of chromosomes. And we only want 1 of each set of chromosomes in our cells, in our gametes, because humans do a sexual life cycle. And sexual life cycle meaning that we combine our genes with other people's to create all of this, diversity that we've got. So it looks a little bit like this. We've got male over here. And when that male is mature, it produces sperm that are haploid, which means they only have 1 of each copy, 1 of each chromosome, some combination. And then through the process of fertilization, that sperm is combined with the haploid egg from a female. So this female produces an egg that is also haploid. And And when sperm and egg meet, we get fertilization. Now, this baby here that is formed, how do we draw a baby? They have a giant head, so there we go. Okay. If this baby is male, all of its cells are now diploid. These cells will divide and differentiate. Until it becomes mature, at which point it starts to produce haploid sperm cells that can be used in fertilization, and the same thing happens here. You get a diploid baby with its giant head compared to its body, and then it grows and matures through meiosis and differentiation till you end up with a sexually mature female, which produces eggs that can combine with the sperm, and so on and so on and so on and so forth. So, you all know the story. So the question that we're going to answer today is, how is it that these sperm and eggs produce haploid cells? The answer is through a process called meiosis. All right, so we are going to now draw meiosis. Meiosis has some very important steps along the way. Let's draw meiosis, and let's make it with a cell that has an N of 3. So this is going to be meiosis. Our cell has an N of 3. Now this whole thing starts off in the exact same way. So we're going to have interphase here first, okay? And in interphase, we're going to have our nucleus right before meiosis starts. Let's draw a little, medium, long, little, medium, long. Okay, so this is interphase before S phase has started. So then we're going to have S phase happen in interphase, and now our nucleus is going to have 2 little black chromosomes, and 2 little red chromosomes, and 2 medium black chromosomes, and 2 medium red chromosomes, and 2 long black chromosomes, and 2 long red chromosomes. This is interphase. After S phase. All right. Let's move down here now to prophase. Now, recall what I drew right over here. And what I drew over here is that these cells are going to divide twice, which means that we are going to have 2 parts of meiosis. We're going to have what we call meiosis 1, and we're going to have what we call meiosis 2. So this isn't just going to be prophase. This is going to be called prophase 1. That's a Roman numeral I for 1 there. This is prophase 1. Now, one of the normal things happens, and that is that the nucleus is going to break apart. And the centrioles are going to move to the other ends of the cell. They're going to start to make their spindle fibers here. But something really unique and different happens right here. Now, let me give you a quick little vocab refresher here. This is my vocab refresher box. I'm going to patent this and trademark it, so if any of you copy of it, you have to give me money. Okay, so if we have 2 chromosomes here, chromosome 1, chromosome 2, and they're the same size, they have genes in the same loci, and so on and so forth, These are called homologous chromosomes. But they're different colors because one is maternal in origin and one is paternal in origin, and so they don't have exactly the same sets of information. Okay? So these are homologous chromosomes. Now, after S phase, we make a couple of exact copies of these things. Now there's a second red one and a second black one. These two black ones are not homologous chromosomes. These two black ones now are what we call sister chromatids. Same with this. These two red ones right here are not homologous chromosomes. Since they are exact copies of one another, they can't be homologous. These are sister chromatids. Now we could consider one black one and red one homologous chromosomes, but we could also consider both of these black ones together and both of these red ones together homologous chromosomes as well, because the black ones contain a unique set of genetic information compared to the red ones, even though they're the same size and have genes in the same places. So it's important that we keep these vocab words in mind when we're talking about prophase because of what's going to happen in our prophase 1 here. So prophase 1. Now what's going to happen is that the pairs of sister chromatids will pair up, just like in mitosis. But now the homologous chromosomes are going to pair up with each other. What does this look like? Well, the two black ones are going to pair up next to each other. Then the two red ones are going to, that are homologous, are going to join right up right next to it. I want you to notice that I overlapped those right there a little bit down there on the bottom. This is really, really, really important. That pairing up right there, where one overlaps the other one, is called crossing over. And in crossing over, some of the DNA from one of the chromosomes actually loops in and joins with some of the DNA from the other chromosome. This is a really important process, and this is the process that the cell uses to make sure that chromosome 1 is paired up with a chromosome 1, and a chromosome 2 is paired up with a chromosome 2, and a chromosome 3 is paired up with a chromosome 3, and so on and so forth. Okay? This crossing over is incredibly important. Now, we need a vocab word to be thrown in here. And that vocab word is chiasmata. God, that washing machine is loud. A chiasmata is the place where the crossing over has happened. So if I was to zoom in really close on mine here, my chiasmata would be right here. Chiasmata are where these chromosomes have joined together. I didn't write that definition down. You might want to pause the video and record that. But the chiasmata is where the homologous chromosomes are held together. Okay. Now this whole set of 4 together, this whole set of 4 chromosomes all together is called a tetrad. Tetra meaning 4. Think about the game Tetris. In the game Tetris, all of the game pieces are made of 4 squares in different arrangements. Alright, but let's draw our tetrads for our other ones, because we've also got medium chromosomes and long chromosomes as well. So our medium chromosomes, the sister chromatids, will join together, and then they will cross over and form a chiasmata with the red sister chromatids as well. So now we have a pair of homologous chromosomes together because the blacks and reds are together. And the same will happen with our long ones. The sister chromatids will connect, The other sister chromatids connect, and then they will join together right there through crossing over and form a chiasmata. Now, there's a really interesting little piece of genetic recombination that happens right here. And sometimes the crossing over results in recombination, which means that sometimes the chromosomes will actually switch little chunks of DNA. So how might this look? It might look like this. If we take our two chromosomes here and we cross them over right here, sometimes in this area right here, little pieces of DNA will actually switch. And what we end up with is a black chromosome that has a little piece of red on it, and a red chromosome that now has a little piece of black on it. And now imagine that you inherit, imagine that this is you now, and imagine that you inherit this chromosome right here. It's no longer just a maternal chromosome. It now is a maternal chromosome with a little piece of paternal DNA on it. This right here is, I'm going to call this star 1, because this is our first place where in meiosis we create new variations. And on these chromosomes, of course, are little genes. And what if this new combination of genes, what if this maternal gene combined with this maternal gene here produces a better combination than everything else? Oh my gosh, that's so amazing. And now they're running the garbage disposal upstairs. Everybody can be quiet. Okay, anyway. So this is spot number 1 where we get like crazy amounts of genetic variation because of meiosis. All right, let's do meiosis 2. Now, prophase 1 and metaphase 1. Metaphase is what I meant to say, not meiosis 2. Prophase 1 and metaphase 1 are the longest ones for me to talk about. It's not going to be this long for every single thing. So we should be able to zoom along a little bit after I have to talk a little bit more detail about metaphase 1 here. So that should be in blue. All right. So this is metaphase 1. So in metaphase 1, you know what happens, okay? And that is that all of these sets of tetrads are going to line up along the equator of the cell. The centrioles will have created spindle fibers that have latched onto the centromeres of each pair of sister chromatids. So let's draw these here. Here I'm going to put my medium one on the top. Still looping the bottom over, still showing that crossing over. I'm going to put my little ones right here in the middle. Don't forget to overlap the middle ones a little bit to show that crossing over. Now, for this bottom one down here, I'm going to show the recombination on just one of these, okay? And in your drawing, you should show recombination on one of them as well. All right, let's get my spindle fibers connected in here. All right. Now, there's another spot where a lot of recombination happens right here. A lot of recombination happens right here. And this is a spot that in meiosis where what's called independent assortment happens. Now what I mean by that is I want you to notice that I drew all the black chromosomes on one side here, and I drew all the red chromosomes on one side here. Your cell doesn't know the difference between the maternal and paternal chromosomes. All your cell does is it just lines the chromosomes up in the middle. Your cell does absolutely zero differentiation. Absolutely zero differentiation. Right here. And this is only one way that metaphase could have happened. Another way that metaphase could have happened is that I could have drawn my chromosomes like this instead. I could have put 2 black on the top and then 1 red on the left, but that's not it. I could have also drawn it like this. I could have also made it so that each side had 2 and 1 of each. I could have also made it so that all the red were on the left and all the black were on the right. There's a whole bunch of possibilities. And this random orientation of which side the maternals and paternals go on create new variations. How does that work? Well, if this is the egg cell that's made, that egg cell receives all black chromosomes, and in this case the black ones we'll say are the maternal ones, and this egg cell would get all of the red ones, and in this case that means paternal. And so this egg cell would contain all paternal chromosomes, and this egg cell would contain all maternal chromosomes. That's interesting. But if you do it right here, this egg cell would contain 2 maternal and 1 paternal, which is a new combination. It's a new orientation. We can put a number to this. Since there's 2 of each chromosome, we can raise it to the n power, which is the number of unique ones, and we can see that in an n of 3, there are 8 possible arrangements of maternal and paternal chromosomes on each side of the cell. Now imagine with humans, our n is 23. 2 to the 23rd, that's a big number. Everybody pause for a minute and try to calculate that. There are 2 to the 23rd is 8,388,608 possible combinations of just maternal and paternal chromosomes. That doesn't even include the variations that we get with crossing over. All right, that was metaphase 1. Like I said, the rest of this is going to be pretty quick. Let's draw anaphase 1. Writing is a problem for me, apparently. So in anaphase 1, those spindle fibers are going to shorten, which will then separate the homologous chromosomes from one another, but the paired sister chromatids are going to stay together. Don't forget to draw your crossing over chromosome as being crossed over. This is the hardest part of this whole thing, is always remembering to keep those pairs together. Alright, that takes us down to telophase 1. Telophase 1, our cell starts to divide. We get a nucleus that reforms around our chromosomes. Whoops. There's no centrioles in the nucleus. Stupid Kish. Alright. So we've got our medium, we've got our small, we've got our long one that has our little recombined piece of DNA on it. Medium, little. Okay. Okay, now cytokinesis 1 is going to happen. And our two cells will actually divide now. We'll go ahead and keep them all paired up right next to each other here. Whoops, wrong colors. Let's just erase that boy. All right. All right, we need to have two spindle fibers now. I'm sorry, not two spindle fibers, two centrioles. Okay. God, that washing machine. I hope you guys can't hear that. It's embarrassing. All right. Prophase 1 happens now. Okay, so now from here on out, everything is going to happen with both of the cells that have been made. Okay, so this is going to get just a little chaotic, so I am going to make sure to put, it should have been yellow. I am going to toss in some yellow lines just to separate my two cells from one another. If you're just going straight up and down in your notebooks, like that shouldn't be a problem. All right, I'm going to eat a banana. Let's draw prophase 1. All right, first of all, this is prophase 2. Made that mistake there. Okay, prophase 2, you can see I drew my nucleus breaking apart. My spindle fibers started to come out of my centrioles. Don't forget that your chromosomes here need to still have that recombined crossed over part on it. All right, how did you do? Hopefully it looks just like mine. I'm going to pause my video in just a second, and I am going to draw metaphase 2 now. You're going to draw metaphase 2 for each of these. You should know what to do. You should know what this looks like. Have you been following along in the videos? I'm going to draw a yellow line across mine just so that you don't get confused. But this metaphase 2 is happening for each of the cells. Each of these cells are going through this second round of division here. All right, I'm going to pause my video, go ahead and draw metaphase 2, and then we'll come back and check it out. All right, here's my metaphase 2. The spindle fibers have pushed and pulled the chromosomes to the middle after connecting to their centromeres. Don't forget to draw your recombined chromosomes there. All right, draw my yellow line. Now, each one of these cells is going to go through anaphase 2. All right, go ahead and draw yours and then come back and check. All right, so check yourself. How did we do? Okay. First question I want to ask you is, did the homologous chromosomes separate here, or did the sister chromatodes separate here? Pause for a minute for you to think about that. Go over to the patented vocab refresher box if you need to. All right, good job, Bashara. Yeah, it was the sister chromatids, all right? Okay, I'm going to draw another yellow line. Everybody draw these cells now. Having finished up anaphase, draw each of these cells going through telophase 2 now. Awesome. So once again, key point here is remember to keep that one particular chromosome as having recombined. Looks like I did the wrong one on mine. Should have been that one right there. This one should be completely red. Okay, there we go. All right. Now notice how there's only one of each chromosome now. Each nucleus that is forming has one medium, one short, one long. One medium, one short, one long. One medium, one short, one long. Okay. Here comes my yellow line. Can anybody guess what the last one is to come here? What's our last one to come? All right, good job, Amira. This is cytokinesis 2. Cytokinesis 2. So I'm going to pause my video. You pause yours. Great. Okay. Here are our 4. Count them. 1, 2, 3, 4 haploid gametes. Now for simplicity's version, these would become 4 egg cells or 4 sperm cells or something like that. And next year you're going to learn a little bit more detail about how these cells form. The processes are called oogenesis, with the OO being like a Latin root for the egg, and spermatogenesis. And so it happens just a little bit differently. But what you just did is meiosis kind of overall as a whole. Now, there's one last thing to note here. And that one last thing to note is way over here. And that is our star 3. You can't choose which sperm or which egg gets donated. And so, therefore, you can't choose which combination of maternal and paternal chromosomes you are going to pass on. And as a result, it's a total random combination. And so, this third spot here of increased genetic variation happens right here at this spot, which is random fertilization. And I'm not writing it down. You need to pause this and you need to record what I say here, or write down what I say. And that is that you can't choose which sperm fertilizes which egg. And as a result, that fertilization is very random as well. So in meiosis, there are 3 places that genetic variation increases. And this is so incredibly important because we don't want to produce the exact copies of ourselves. If we produce the exact copies of ourselves and our environment changes, it's entirely possible that our environment, this new environment, is not a successful place for our versions of our chromosomes to be. And as a result, populations and species could die out. This is how evolution works. And so as a result of meiosis and this sexual reproduction and this random orientation and the crossing over and the random fertilization, we end up with these new combinations of genes and chromosomes. And some of these are better suited for newer environments than others. Now, we do have one last thing to talk about, but we are going to save that for Wednesday's notes, and that is how errors in meiosis can lead to things like Down syndrome. Actually, you know what? That is going to be the second part of today's activity, because we really want all of Wednesday to focus on how understanding meiosis can allow us to do Punnett squares. Yay! Okay, everybody, take it easy.